this is, I'm sure, a very familiar image to all of you, an image of the human brain. And as we can all appreciate that this human brain is composed of very complex components, and these complex components are continuously interacting with each other. We know that actually the most rapid growth of the brain happens in the several years after birth. So here's a chart of brain weight versus age, and we can see that only after 3 years following birth, the brain size rapidly grows and reaches its about 90% of the size of an adult brain. And what we know that during this process, it's not that the brain is making more cells, because that happens very early on during development, but because more synapses, more dendrites, and more connectivity is forming in this brain. So synapses, we know that synapses are very important sites because they are the sites where learning process happens and memories are stored. And majority of these synapses are generated soon after birth during early development. And finally, in an adult brain, a given neuron has approximately 1,000 synapses. So the human brain is composed of about 10 to the 11 number of neurons, which make up about 10 to the 14 synapses in our brain. And I have, before questions, I have 2 slides that I want to show you. One of the slides is answer to a very common question that I get in these kind of seminars, and that question is, what's happening in my brain? What's going on? You talked about development and babies and early on, but what's going on at my age? And here's a graph showing that. So there are 2 plots here, number of neurons versus number of synapses throughout your lifetime. And we all have, unfortunately, passed the peak of synaptic density in our brains, and we have reached this steady plateau phase where synapse number are steadily decreasing in our brains. Okay, so in our laboratory, we are interested in one transcription factor that is called NeuroD2 and how it functions in development of the brain. So we do this by studying live neurons. So we take a mouse, we extract we take pregnant mice, we extract the embryos, and we isolate neurons from these embryos. So we do this, we isolate neurons from the embryonic brain because we want to catch them at a young age. We want to catch them before they have actually differentiated on to a very mature neuron. Then we put them in culture, we let them grow and differentiate under the conditions that we program them to, and then we analyze these neurons, either by microscopy or by other molecular biology techniques. Our main question is focused on understanding the genes that NeuroD2 is regulating in the cell. We do the different kinds of experiments. For example, we look at gene expression analysis, comparing normal neurons with neurons where we get rid of NeuroD2 function. And we also try to identify direct binding targets of NeuroD2 on DNA molecules by doing techniques such as chromatin immunoprecipitation. And by the intersection of these data sets, we are forming models of how NeuroD2 regulates neuronal differentiation. And finally, here are our laboratories that we carry out our research, and the students who help care and who do this work firsthand, Semra, Efil, Gökhan, Cansu, and Gizem. And we are thankful to our funding sources. And thank you.